Hey guys, welcome back. We're at concept 23.3. It says natural selection, genetic drift, and gene flow can alter allele frequencies in a population. We have been talking about populations that are at this theoretical Hardy-Weinberg equilibrium, and we said that that's really kind of a theoretical construct. It's kind of a tool. We can use that in the same vein as saying, assume no frictional effects when we're doing physics problems. We rarely see natural populations at Hardy-Weinberg equilibrium. Because populations are always evolving to some degree because of various circumstances in their environment, and we're going to look at those circumstances right now. So this is actually the second time I've recorded this and the next chapter portion, because the first time I recorded it, I did the whole recording and then went to check the recording and upload it to Schoology, and I realized that I had turned my mic off and had left it off the entire time I was recording. So this is the second round of concept 23.3 for me, and the first round with audio for you. It says natural selection, genetic drift, and gene flow can alter allele frequencies in a population, and we're going to look at all three of those, and we're going to find out that natural selection is the primary mover and shaker when it comes to adaptive evolution, but genetic drift and gene flow are both important, and they also will affect the various allele frequencies in a population. So we have already talked about a few other things that can affect allele frequencies, such as mutations. Brand new mutations pop up on a regular basis. That violates condition 1 of Hardy-Weinberg, and they can alter allele frequency, but mutations are still fairly rare. You wouldn't call that a very common occurrence, and so the change from one generation to the next is likely to be very small, although mutations obviously can have a powerful effect on a particular individual and over time have a great influence on a population. Then we can also talk about non-random mating, which is a violation of condition 2 of Hardy-Weinberg. Non-random mating can also affect the frequency of alleles, particularly the frequency of homozygous and heterozygous genotypes, but by itself, non-random mating is not really a major mover in having a huge effect on the overall allele frequency in the gene pool. It may affect how many homozygous dominants and how many heterozygous and that type of thing, but remember, we're throwing all the alleles into one bucket, and it really doesn't do much to change the allele frequency within that entire bucket. So we're going to look at the ones that really do have a major impact, and that would be these three: natural selection, genetic drift, and gene flow. First, let's do a really quick intro into all three, and then we'll kind of dissect them and go into more detail. Natural selection we've already discussed. Differential success in reproduction results in certain alleles being passed to the next generation in greater proportions. This causes what we call adaptive evolution, to where the creature becomes better matched to its environment. And a good example they use in the textbook is a particular allele that can be found in fruit flies that confers a resistance to DDT. DDT is a powerful insecticide, or it used to be a powerful insecticide, and it was used quite extensively back in the 1950s and 60s. It does have some other side effects in the environment, so it was eventually banned, but it was used quite a bit earlier, before you and I were born. So there is an allele in some fruit flies that causes them to have some resistance to DDT. And after DDT began to be used quite extensively, that allele was selected for via natural selection, and it became quite a bit more frequent in the fruit fly population. The way that they kind of found this, or the way that they compared populations, was they looked at laboratory strains of fruit flies, D. melanogaster. These laboratory strains had been collected from the wild way back in the 1930s, and since that time had been grown in the laboratory separate from the natural environment. And so back in the 1930s they didn't have DDT, so these original populations that were brought into the laboratory had never been exposed to DDT, and the percentage of the DDT resistant allele in these fruit flies was 0% of the lab raised fruit flies had this allele. But when they compared that to fruit flies that were caught out in the wild in the 1960s, which is about 20 years after DDT was introduced, the allele frequency in the wild populations was about 37%. So just in a matter of, say, 30 or 40 years, this allele frequency went from 0 to 37% found in the population, and that's a direct result of natural selection. I think we already mentioned how natural selection is not a random part of evolution, but here genetic drift is a more random variation of allele frequency change. Genetic drift really affects smaller populations. The smaller the sample size, the greater the chance of a deviation from a predicted result. If you had a quarter and you flipped that quarter 1,000 times and you got 700 heads, you might be suspicious of that quarter. You might think it's fixed in some way, because that would be very odd. But if you took a quarter and you flipped it only 10 times and you got 7 heads, that still is odd. You would expect a 50-50 ratio, but it's not nearly as odd as 700 out of 1,000. The smaller the sample size, the greater the chance that you're going to have variety and deviation from the norm. So genetic drift is kind of the application of this idea on a population's genetics, where this describes how allele frequencies can fluctuate unpredictably from one generation to the next. This is kind of a random fluctuation, and it tends to reduce genetic variation through the loss of alleles. There is a short little animation here you can take a look at. Right click and hit play, and it talks about genetic drift and it talks about natural selection both, and it kind of gives you an overview of them. So let's continue looking at genetic drift. Again, small populations are greatly affected by this chance occurrence, basically who happens to mate or who happens to survive. So here we have a fairly small population of flowers, and you see there some of them have homozygous dominant genotypes, that would be the red flowers. Some of them are heterozygous, and it's an incomplete dominance, so they have the pink flowers. And in this population we only have one homozygous recessive. So it's a fairly small population. You can look at the allele frequencies, and the P frequency is 0.7 and the Q is 0.3. Now let's say that 5 of these plants leave offspring. Just random chance as to which seeds land in the right spot and are able to grow or don't get eaten by birds or something like that. So let's say just by chance the 5 that are in the white squares, they are the ones that are able to produce seeds and leave offspring. And the next generation, because of the genetics found in these 5, has quite a bit of a different allele frequency. So in this generation the population looks quite a bit different, and now if you counted up all the alleles, the frequency is 50-50, and that's quite a change from 0.7.3 to 0.5.5. And then once again, just by random chance, let's say that only 2 of the plants in the next year leave offspring, and it happens to be these 2 that you see in the white square. If that's the case, well then we have an even crazier, wider swing, to where now in generation 3 we have a P of 1.0 and a Q of 0. So just in 3 generations you can see how the allele frequency swings pretty wildly all over the place. And now from this point forward, if this is our population, we're only going to have the P allele. We're not going to have any Qs at all. It's like it suddenly just disappeared. So in a small population like this, chance occurrences can have a great effect on the gene pool, and we call this genetic drift. There's a couple of varieties of genetic drift that we can kind of take a look at. One of them is called the founder effect. Founder, like the founding father of maybe the small town that you live in or grew up in. A founder would be like the pioneer, the first to arrive. So the founder effect occurs when a few individuals become isolated from a larger population. Let's say there's a hurricane and maybe one small type of bird. 3 or 4 of these birds get blown away from the mainland, and they find themselves on a little isolated island somewhere out a couple of miles off the coast. Well, they would be considered founders, and because you don't really know which birds of the population got blown out there, it's just random chance, they may not be representative of the entire population back on the mainland, so the allele frequencies in that little small founder population can be quite different from what you would find on the mainland. That's a good example of genetic drift, and where the allele frequencies can swing and change pretty wildly in a short amount of time. Another good example of that is called the bottleneck effect. The bottleneck effect is where a fairly large population is reduced in size suddenly, usually by some sort of natural disaster. Maybe it was a flood or famine or wildfire, something like that. Earlier in the chapter, it was talking about those particular birds on the island of Daphne Major, and how after a long, prolonged drought, that there was over 1,000 birds to begin with, and in the end a few hundred survived. That would be a good example of a bottleneck effect, to where the population was reduced quite a bit, and the gene pool of the population that gets left behind may not be very reflective of the original population's gene pool. So if the population stays small after this major disaster, you will have even further genetic drift, and the gene pool may look even more different than it did in the beginning. So here's a kind of an example using a bottle of colored marbles. So this is the original population. You see primarily some blue and then a little bit lesser white and a few yellow. And let's say there's some sort of bottlenecking event that very much reduces the population. Well, what you end up with is a surviving population that may not look similar to the original population. Here you don't have any yellow marbles at all, and the ratio of blue to white looks different than it did over here. So this is a good example of genetic drift caused by some sort of catastrophic event that leaves behind a smaller population than what you began with. Usually the bottleneck effect is caused by some type of natural disaster or major environmental change, but actually humans have such an impact on the environment now that humans can be the cause of this bottleneck effect. So understanding this bottleneck effect helps increase the understanding of how our activity affects the species and the environment that those species live in. A good example in the textbook is this particular bird, this land bird called the greater prairie chicken. And this greater prairie chicken lived throughout Illinois and Kansas and Nebraska in the grassland areas. But in Illinois, it did have a large population at one time, and now its population is very, very tiny because we have reduced its prairie habitat by a tremendous amount there in Illinois. So the population size was cut tremendously, and that reduced the variety within their gene pool. That reduced the they had low genetic variation, and that causes some problems. You start to get a lot of inbreeding, and the egg hatch rate of those birds dropped to about 50%. They give you a nice graphic in the textbook here. Here you have the state of Illinois, and back in 1820 you can see these greater prairie chicken, the range covered almost all the state. And then in 1993 there's only a few tiny little spots of this greater prairie chicken left, and so there was a drastic reduction in population, and there was a correlation to some problems because of that reduction. So here in Illinois you can see the population size, how much it dropped, and you can see the number of alleles per locus also dropped, so the genetic diversity dropped, and the percentage of eggs hatching dropped as well. Compare that to Kansas and Nebraska, where they didn't have this major bottleneck, and the population size stayed more constant, the number of alleles per locus stayed constant, and the hatch rate was quite a bit higher. So the researchers, what they did is they compared DNA from museum specimens that were collected many, many years ago, and they compared the DNA of these stuffed birds, these museum specimens, to the genetic variation they found in the wild herds or the wild flocks of these birds, and they found that there wasn't nearly as many possibilities, not nearly as many alleles in the wild birds as there were in the older stuffed birds that they pulled out of the museums. So they showed a loss of alleles at several loci. To solve this problem, they had to basically bring greater prairie chickens from outside areas and bring them into the state of Illinois and kind of put them in with the current populations. So that would introduce new alleles, and then that increased the egg hatch rate back up to about 90%. It increased the gene pool size, increased the genetic diversity, and that usually brings about good results. You're going to see in a minute, you would kind of think of that as forced gene flow. Gene flow is how animals or creatures move from one population to another and bring their genetics with them. So this was kind of like the researchers forcing gene flow to happen to try to improve the situation for this particular animal. So here's a summary of genetic drift. Genetic drift is a bit of a random process. It can usually is negative because it usually ends up reducing the number of alleles in a particular loci. But number one, genetic drift is significant, mainly in smaller populations. The smaller the population, the more genetic drift you're going to see. Number two, genetic drift causes allele frequencies to change at random. Number 3, genetic drift can lead to a loss of genetic variation within a population. That's usually what ends up happening. Number 4, genetic drift can cause harmful alleles to become fixed in place because you don't have the genetic variation to kind of take care of those and to allow natural selection to occur. So genetic drift is a good example of the role that chance does play in genetic variation. So there are portions of evolution that you would say are random or up to chance, and then there are portions that are not. The next thing we're going to look at that can affect allele frequency within a population and can affect the genetic pool of that population is something called gene flow. Gene flow consists of the movement of alleles among populations. Alleles are transferred through the movement of fertile individuals or gametes. So when, say, birds fly from one area to another, or even human beings as they move around the planet. Now, we can even talk about plants, because plants of course can't get up and move around, but their pollen can be spread by the wind. So you have gene flow, you have alleles that are transferred from one area to another, from one population to another. Gene flow tends to reduce variation among populations over time. If you had two separate populations of a particular bird on an island and they were able to begin mixing together and breeding, interbreeding, over time, those two populations would probably end up just becoming one giant genetic pool, and there wouldn't be a lot of variety between those two populations anymore. They would probably be considered a single population with a common gene pool. Gene flow doesn't consistently lead to adaptive evolution, and that's because gene flow can sometimes be positive and sometimes be negative. So in this case, they're going to talk about how gene flow can decrease the fitness of a particular population. And in the book, they talk about this particular type of bird called the great tit, and it lives on the Dutch island of Vlieland. Man, I don't even know how to pronounce this, Vlieland. And there are two populations on this island. So rather than go over the words here, because I think it's kind of confusing, let me show you the picture and I'll describe what's going on. So here in the little map, we show the island that we're talking about, and here are the two populations. We have one called the central population and one called the eastern population of this little bird, cute little guy right here. Now what you don't see on the map is the mainland, which is off of the map somewhere, and some of these birds also live on the mainland. Now if you compare the birds that live on the island, they have certain alleles that make them better suited for living on the island. And the birds that live on the mainland, they sometimes fly out to the island, but the birds that come from the mainland, their genetics do not make them quite as suited for living on the island. Now the birds that fly from the mainland, for whatever reason, they tend to breed with the birds in the central population. And the birds coming from the mainland tend to not breed with the eastern population. Now if we compare these two populations, we see that the central population, that would be these right here, they have a lower survival rate than the ones from the eastern population. And the reason is because the birds flying in from the mainland tend to breed with the central population, and that gene flow is actually bringing in alleles that are perhaps negative for survival on the island. So that is a good example of how gene flow can sometimes reduce the fitness of a population. It's not always the case. Sometimes gene flow can be positive, and we'll look at an example in just a second. But in this case, that movement of alleles from the mainland to the central population is making the overall population do worse than the other population up here, the eastern population. But like I said, gene flow doesn't have to be negative, it can also be positive. And a good example would be insecticide resistance in certain insects, like for example in mosquitoes, because we sometimes use insecticides to target mosquitoes that carry West Nile virus and malaria. Here, even in East Texas, in Tyler, Texas, and perhaps in Waxahachie, West Nile virus is a concern, and so we sometimes have spraying programs where we spray for mosquitoes neighborhood-wide. Well, there are certain mosquitoes that have resistance to the insecticides that we use. Through various genetic mutations, they have alleles that confer resistance, and when these mosquitoes, if they move from one area to another, they carry those alleles with them, and when they start breeding with a different population, they will spread those alleles, and that will spread that resistance. So that's conferring an advantage, and that's where gene flow can increase the fitness of a population. As those mosquitoes come in that have that resistance, they can spread that resistance through the population through breeding. So overall, gene flow can be positive and can be negative, so you wouldn't say that it consistently causes adaptive evolution, because it can have both sides of the coin, positive or negative effects. It kind of depends on the situation. I mentioned that gene flow is important even in a human population standpoint. From a human population standpoint, humans today, of course, move quite freely around the entire globe as compared to, say, a few hundred years ago, whereas maybe, say, 300 or 400 years ago you had more distinct human populations in certain areas. Now, because travel is so relatively easy, we have interbreeding amongst many of the different human populations from around the globe, and what this leads to is an exchange of alleles, an exchange of genetic information between these populations, and so these populations begin to have fewer genetic differences than they did maybe 300, 400, 500 years ago. And so this is an example of gene flow even in the human species. Alright, let's take a break right there. So we've talked about gene flow, we've talked about genetic drift a little bit. Those two don't necessarily lead to adaptive evolution consistently. The next and last portion we're going to talk about is concept 23.4, natural selection, and as a part of that we'll talk about sexual selection. This is the major game player in evolution, is particularly adaptive evolution, changing the species, changing population genetics over time, and having the creature become, or having the population of creatures become better adapted to their environment. Let's take a break. We'll come right back and finish up.